Hi and welcome to the lecture of Bio 110. In this lecture we're going to be covering mitosis and cytokinesis in meiosis. So now, here again are the objectives for this lecture. Please take the time to understand these objectives as they will be helpful to guide you through the studying for the next exam. As I mentioned to you, when the cells have undergone interphase, they're going to enter into the M cycle. Now, to get into the M stage of the cell cycle, you have to activate M-Cdk, and you have three requirements that are needed for that. Number one is the binding of the M cycling to the mitotic CDK. That complex is inactive. Then you're going to have phosphorylation at two different sites. The kinase W1 will phosphorylate in the inhibitory phosphate, and the kinase CAG will phosphorylate in the activating phosphate, and still at that point this complex is going to be inactive. To induce activity, you require the activation of CDC25, which will remove the phosphate in the inhibitory site and therefore leading a molecule that is active. Now, when acid M-Cdk is ready, it will help assemble the Mitotic spindle and induce chromosome condensation. One interesting thing that I would like to point out is the fact that M-Cdk is having a positive feedback loop with CDC25. Active M-Cdk is going to be able to phosphorylate inactive CDC25, allowing it to be activated. At that point, the active CDC25 phosphatase will be able to remove the inhibitory phosphates of inactive M-Cdks, inducing more active M-Cdk. This positive feedback loop will allow the rapid progression into the M phase of the cell cycle. But I mentioned to you also that active M-Cdk can phosphorylate other molecules like the APC, the Anaphase promoting complex. In that case, you have a negative feedback loop where the phosphorylation of the APC molecule will activate it, and therefore it will then target the APC to work and lead the M cycling for destruction by the proteasome. So you have two different feedback loops, one positive feedback loop activating CDC75, so that will lead to more active M-Cdk, and a negative feedback loop inactivating or activating APC, which leads to the destruction of the M cycling. Now, when we look at the M phase, we're going to have two different purposes. The first one is going to be to segregate the replicated chromosomes for each of the daughter cells. And that is going to be performed in 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. The second portion of the M phase is going to be to separate the two daughter cells by cytokinesis. So what we have in this image are the 6 different steps that are required for M phase. As I mentioned, meiosis will include prophase, prometaphase, metaphase, anaphase, and telophase, while the separation of the two daughter cells will be ruled by cytokinesis. And all these processes are going to be controlled by M-Cdk. Now, when we take a look during mitosis, we can stage mitosis by the presence of the spindle position, by the presence and the state of the chromosomes, and the presence and state of the nucleus. So what we're going to see is that when we are taking into consideration the stage of the cell cycle, that we have to look at those 3 points: the spindle, the chromosomes, and the nucleus. The first stage that we're going to be looking at is interphase. And as you know, during interphase, you have the cells that are going to be growing, they're going to be making more organelles, and they're going to be replicating the DNA. At the end of the G2 stage, you're going to have now the cell with its double chromosomes, as well as the duplication of the organelles, including the centrosomes. So what you see here in this image are the duplicated centrosomes that are irradiating macrotubules. Now, the first stage of the M phase is prophase, and in prophase is when you have the initial separation of the centrosomes. So the centrosomes are going to start migrating away from one another, surrounding the nucleus, and you also are going to have the first step of chromosome condensation. So at this point, you can appreciate here in the fluorescence image on the right, you have a cell which has been marked with tubulin GFP, so you can appreciate the macrotubules, and the DNA have been also fluorescently labeled, so you can also see it. You can appreciate that inside the cell, the nucleus is very still intact and all the chromosomes are nicely condensed and visible. And the mitotic spindles, shown here by the yellow arrows, have begun to separate from one another and they're starting to move in opposite directions of the nucleus. In the next step, what we're going to see is Prometaphase. And Prometaphase starts when the nuclear envelope has been abruptly disrupted. That is going to be mediated by the activation of M-Cdk, which is going to phosphorylate the lamins in the nucleus as well as the nuclear pore proteins, therefore separating the nucleus and everything falls apart. That now exposes the chromosomes, which are already condensed, into the cytoplasm, and therefore they can now bind to the macrotubules that are present through their kinetophores and begin to move. So as you can appreciate here in the fluorescent microscope image, you have the two spindles which are opposite to one another, and the chromosomes, they are randomly placed around in the cytoplasm, but they're engaging already the macrotubules. At this point is when motor proteins in the macrotubules will begin to move the chromosomes to the metaphase plate, the center of the cell division, and that is the process of metaphase. In metaphase, the chromosomes are going to be aligned this imaginary plane, which is the equator of the spindle, and that's the metaphase plates. I modified this image from the book and I put a white dashed line, and as you can see, all the chromosomes are now aligned perfectly in the middle, binding to the mitotic spindle macrotubules, but very needle on the metaphase plate. So the chromosomes are bound to the macrotubules because of their kinetochores, and the sister chromatids are getting ready to be separated. That is the process that is going to happen during anaphase. Anaphase begins in the next step, and over there in anaphase you're going to have the separation of sister chromatids that happens synchronously and simultaneously, pulling away the sister chromatids from one another. This will ensure that each independent daughter cell will get the exact same amount of DNA. So at that point, as the chromosomes are moving towards the mitotic spindle is also moving away from one another and getting closer to the opposite poles of the cell. So you will have shortening of kinetochore macrotubules and pulling of the astral macrotubules in the cell. And I'm going to show you that in a moment. The last stage of mitosis is going to be telophase. And in telophase, you have had the complete separation of the sister chromatids performed by the spindle macrotubules. You also have the beginning of the reconstruction of the nucleus. At the same time, you have the formation of a contractile ring made of actin filaments and myosin 2 proteins that are going to begin to assemble in the area of the metaphase plate. In here in the right, you can appreciate a cell that is already in telophase. The chromosomes are completely separated from one another, and you begin to see a dark area in between the two cells. That is the place where the actin molecules, which are not labeled in this image, are beginning to obscure the tubulin GFP, and therefore they form a line. So you can appreciate that right in the middle. The last part is going to be cytokinesis, and cytokinesis is going to be the actual separation of the two cells into independent daughter cells. What you have here, it's the compaction of the mitotic, of the contractile ring, creating a cleavage furrow. As you can appreciate in the image on the left, the macrotubules that are coming from each pole are still present in the middle, and eventually they will be guillotined and cleave away. What you can see here from the right is that you have that area of darkness in between the two cells as the contractile ring begins to contract more and more, and eventually separating the cells into two daughter cells. So you have a really tight area of around the cleavage furrow. You also have the separation and the reformation on the interface array of macrotubules around a nucleated centrosome. So that is mitosis in a nutshell. So what I would like you to do is to be able to know and understand the difference between each of the stages of mitosis and cytokinesis. And what I'm going to do now is to show you some of the details that are interesting about the process. So now, before the cell enters into N phase, two things need to happen. Number one, the DNA needs to be replicated completely, and that is going to happen during S phase. Number two, the centrosomes need to be duplicated, and that's going to be happening during G1 and G2. So one cell that has now been able to have its DNA replicated and centrosomes replicated will enter into N phase. So in this slide, what I want to show you is the process of centrosome replication. Here in G1, after cells have divided, you have a centrosome that contains two centrioles. They are surrounded by a centrosome matrix. Late during G1, the centrioles in that structure begin to separate, and they separate sufficiently to have some distance between one another, and that now during S phase allow for the generation of a daughter centrosome 90 degrees away from the other. So now both of the original mother centrosomes are having a daughter centrosomes generated. At the end of G2, the cell would have completely duplicated the centrosomes, and those centrosomes will still be surrounded by a common centrosome matrix. And during N phase, those centrosomes will separate into independent mitotic spindles. Another thing that is going to change is going to be the state of the chromosomes in the cell. In this image, what you can see are the chromosomes in a cell in interphase in G1. And as you can see, the chromosomes are compacted but not condensed. On the left-hand side, you are looking at mitotic chromosomes which have gone through condensation. What you're going to see are two chromosomes that are attached to a common centromere, and each of those sister chromatids, it's an identical copy of the other. So this is mediated by M-Cdk activity. One of the events that is going to happen during the S phase is the duplication of DNA. So the copied sister chromatids need to be held together in order for the cells to know which chromosome is which, and that is mediated by the action of proteins called cohesins. So cohesins will maintain the sister chromatids close to one another before condensation. Now, when M-Cdk becomes activated, it will induce condensing proteins to mediate the condensation and the resolution of the sister chromatids. As you can see in the image, the condensing rings, which are wrapped around compacted DNA, are going to form structures that eventually will lead to the formation of the mitotic chromosome. And the mitotic chromosome, as you can see here in this electron micrograph on the right, contains two different sister chromatids which are held together in a centromere. Now, during G1 and SG2, you're going to have the duplication of the centrosomes. So at the point that the centrosomes gets divided, at S and G2, you eventually are going to enter into prophase, and in prophase you begin to have the condensation of the chromosomes and the separation and assembly of the mitotic spindle. Once the chromosomes have been condensed during prometaphase, you're going to eventually get the destruction of the nuclear envelope, allowing the proteins of the mitotic spindle to interact with the chromosomes through their kinetochores. Eventually, those chromosomes will be put in the middle of the metaphase plate to allow them to be segregated during anaphase. Now, two different centrosomes are going to be generated during the last phase of interphase. And therefore, those two chimeras are going to have independent generation of microtubules, which are growing the microtubules through dynamic instability. So those microtubules are going to be growing in all directions. Eventually, some of those microtubules from each of those poles are going to interact one another, and they are going to overlap. And at that point, you will have proteins that are going to held them in place. So the ones in the middle that are interacting with one another and held in between the spindle poles are called interpolar microtubules. And the ones which are radiating from the other sides of the spindle poles are called aster microtubules. So here is a slide that is describing the different kinds of proteins that are interacting between the interpolar microtubules. So the first thing that we're going to have are dynein motor proteins that are bound to the membrane. And those dynein proteins are going to, as you know, walk the microtubule towards the negative pole of the complex. And therefore, since they are now held together in the membrane area, they are not able to move. So what they're effectively doing is pulling the centrosome towards them. So that is one of the forces that you have, the pulling of the mitotic spindle towards the membrane by the dynins. On the other hand, in the spindle microtubules that are in the interspindle zone, you have kinesin 5 motors. Kinesin 5 motors have two active sites, and therefore they can bind microtubules that are coming from the left centrosome and from the right centrosome. Since they're pulling towards the positive end, what you have in effectiveness, it is a separation of the centrosomes from one another. So kinesin 5 is able to push the centrosomes away from one another. Kinesin 14 is a special kinesin because it is the only kinesin that migrates towards the negative pole. And as you can appreciate from the slide, kinesin 14 is covalently bound to a macrotubule, and it's using the macrotubule from the left centrosome as a railroad track to walk, and therefore it's walking towards the negative pole. In doing so, kinesin 14 it's going to be tightening the distance between the left centrosome and the right centrosome. Last but not least, what you have are the work of kinesin 4 and kinesin 10. Those are able to bind to the chromosome arms of the sister chromatids and move them during prometaphase to the center of the metaphase plate, So what you have at the end are the forces of kinesin 5, which is pushing the macrotubules centrosomes, away from one another. You have the effects of dynins in the opposite point of the cell, which are pulling the centrosomes towards them, and you have the effect of kinesin 14, which is trying to bring together the centrosomes. So exactly how those three forces working together achieves the preparation of the cells to undergo anaphase is not well understood, but what we understand is probably that it is all three forces are required to the separation of the chromosomes through anaphase, as well as the separation of the spindles during cell division. Now, let's take a look at what's happening in prometaphase. As you know, M-Cdk is active during the M cycle, and M-Cdk is going to be able to take and phosphorylate the lamins within the nuclear lamina, as well as the nuclear pore proteins, inducing them to disassemble. So that is going to be an important step in disassembling the nucleus. Now, the condensed chromosomes, which are present inside the nucleus, are in the cytoplasm, and they can engage the macrotubules between the spindle poles. When you have the macrotubules in the spindle poles, they're going to have two different kinds of interactions. The first one is going to be the aster macrotubules, shown here in green. And the aster macrotubules, as I mentioned to you, are radiating away from the centrosome, from the spindle pole, and interacting with dynein proteins which are present in the inner leaflet of the membrane. They're used to pull the spindle poles towards the membrane. In pink, what we have are the interpolar macrotubules. Macrotubules which are associated with one another in between the two spindle poles. And as I mentioned, proteins like kinesin 14 and kinesin 5 are working to hold these molecules together in place. Last, what you have are the kinetochore macrotubules shown in blue. And the kinetochore macrotubules are macrotubules that are binding to the kinetochore complex next to the centromeres in the chromosome. Let's take a look to see how those macrotubules are interacting. One thing that I want to point to you is that chromosome post division in S phase, it's going to be composed of two independent chromatids. So we have two different sister chromatids held together, and you will have then the macrotubules from each of the spindle poles shown here in green binding together to the centromere region of the chromosome through the kinetochores. So once the nuclear envelope is gone, the mitotic spindle can access the chromosomes to attach them. So this is the region here shown in red in the illustration. So the kinetochores are a protein structure that is able to bind independent macrotubules by having a color structure. So the color structure is going to allow a macrotubule to thread through it like a piece of thread can go through the eye of a needle. As you can appreciate from the electron micrograph, this is a chromosome that is in anaphase. You can see the chromatid that is bending over because it's being pulled towards the right. In the middle portion, you have a kinetochore, and what you have is a large array of macrotubules which are embedded in the chromosome in the area of the kinetochore, helping pull the chromosomes toward the right. So in order to separate those sister chromatids, which are held together by cohesive rings, you have the activation of the anaphase promoting complex. As I mentioned earlier, the anaphase promoting complex can be activated by M-Cdk. APC, the anaphase promoting complex, it's a molecule that is going to promote the ubiquination of proteins. So once activated by M-Cdk, the APC is able to bind to a protein called securin. Securin is a protein that is inactivating a protease called separase. So during the cell cycle, separase is being kept inactive by binding to securin. Now that the sister chromatids need to be separated, the APC is able to target the securin molecule for degradation, and you have the activation of the enzyme separase. Separase will be able to digest the cohesin rings, and once the cohesin rings are digested, that will allow the separation of the sister chromatids from one another during anaphase. This is why we understand that APC is the anaphase promoting complex. So there are different forces that are working for the separation of the macrotubules during anaphase. So when the polarization occurs, you have the protofilaments in the macrotubule which are curling outwards. As you remember, this is part of the catastrophic event. But because the color structure of the kinetochore it's believed to be wrapped around the macrotubules, that force that is forcing the macrotubule to separate, it's being used and kept in place by the kinetochore color structure. And therefore, the kinetochore is being pulled along the macrotubule towards the negative pole as the macrotubule is undergoing a catastrophic event. So in principle, that is moving it to the negative end. And this is an ATP independent event. This can happen in vitro in the absence of ATP. So it's believed that the energy required to move the kinetochore in part is coming from the actual depolarization of the macrotubule, which already spent energy by hydrolyzing GTP to be assembled. So let's take a look at the two forces that are moving the chromosome, towards the end of the poles. We have two different forces. The first one is going to be the chromosomes being pulled towards the poles. And that is believed to be happening, shown here in A, during the stage of Anaphase A through the shortening of the kinetochore macrotubules. As I mentioned to you earlier, after the kinetochore macrotubules are being shortened, they are going to be able to pull the chromosomes along with them. So that is going to generate the force needed to move the chromosomes towards the spindle pole. That is part A in Anaphase A. During Anaphase B, you have a second set of forces, and these are going to be the forces that are going to be pushing and pulling the macrotubules. So force number one, it's going to be the sliding force, and the sliding force is being generated most likely by kinesin 5 when it is pushing the macrotubules, interpolar macrotubules, away from one another. And the other part that you have is the pulling force, mediated by the astral macrotubules and dynein, and that is going then to be pulling the poles towards the cell cortex. It is by the membrane. So the combined forces when the kinetochore macrotubules are falling apart and therefore they are moving the chromosome towards the negative A during Anaphase A, together with the poles being pushed and pulled together by the action of dynein and kinesin 5, will eventually lead to the separation of the spindle poles and the chromosomes during anaphase. After anaphase is complete, you will have the cell begin to go through telophase. As I mentioned to you, one of the main activation of during the M cycle is M cycling M-Cdk, and M-Cdk is going to activate APC. But APC, one of the targets is going to be M-cycling. So M-cycling will be targeted for the structure, which is going to inhibit CDK activity during anaphase. This is going to now allow the activation of phosphatases that will be able to remove the phosphates from the targets that CDK phosphorylated. One of those proteins in yeast is the protein CDC14. Once the phosphate, for example, has been removed from the lamins and from the nuclear pore proteins, those will be able to begin assembling one another, and eventually they start to interact with the daughter chromosomes. And at that point, what you have is the formation of the nucleus during telophase. Eventually, those vesicles containing membrane in the bound to the nuclear lamins are going to fuse, creating an envelope that eventually completely reforms the nucleus. At the point of the end of telophase, what we have is the beginning of cytokinesis. And as this image is showing you in the top, you can appreciate that the two cells are beginning to separate from one another, and that you have the formation of a cleavage furrow between the two of them. When you take a look at the cleavage furrow, what you have is the formation of a cytoskeletal structure made of actin and myosin II, and that is called the contractile ring. The contractile ring is going to be using ATP to eventually separate and cleave off both cells from one another. So the remaining interpolar macrotubules in the middle will be cleaved by the force of the contractile ring, and eventually that will pinch the membranes off between both cells, creating now two daughter cells. What you can see from this fluorescent microscope picture are the location of the cleavage furrow and myosin II. So in orange in the middle, you have the myosin II, and as a control staining, you have myosin I, and you can see that myosin I, it's working in the cortex in the opposite side, helping move the cortex away from the center, and you have myosin II with actin in the middle during the cleavage furrow, helping the cell undergo cytokinesis. So one line of evidence that helped us understand that was to create myosin mutants during cell division. So this is illustrated here in the bottom right. So here you have a cell that has undergone cell division, and in the control untreated sample, what you have is the generation of independent cells from one independent original mother cell. But in the right, you have a cell that either has been deleted for myosin 2 or injected with an antibody that can inhibit myosin 2 function. And at that point, you will see that that cell has multiple nucleus because the cell cycle has continued, but cytokinesis is being inhibited. So you have one very large cell that has been generated with many different nucleus inside because the stage of cytokinesis never happened. This was evidence that myosin 2 and the acting cytoskeleton was required to induce cytokinesis. So what we have shown here is that the stages of cell cycle will mediate the function of the cell, allowing the cell to divide into two independent daughter cells. So like I said, to summarize, number one, you have the formation of the pre-replication complexes assembling at the origin, and that is happening in the early stages of G1. You have activation of different cycling CDKs that are going to allow the cell to go through the cells at the G1 stage of the cell cycle. Eventually, you have DNA replication in the S phase by the activation of S phase CDK that will mediate the duplication of DNA. Eventually, after the cell has duplicated, you will go through another phase of growth in the G2 stage, and that will lead you to the M phase. And during M phase, you have the increased activity of the M-Cdk, which is going to allow the cell to undergo mitosis and cytokinesis. So important proteins to remember are going to be the M-Cdk and of course the APC protosome that is going to help degrade molecules allowing anaphase to continue. At the end, the two daughter cells are going to be able to be separated through cytokinesis. Now, let's take a look at a parallel system, which is meiosis. So, meiosis, it's the process in which we generate gametes. In this image, what we have is an egg that is about to be fertilized by some sperm. So both the egg and the sperm only have half the DNA information than a somatic cell has. So to generate the cells with less genetic information, the germ cells in the gonads are going to undergo meiosis. Now, all the somatic cells that we have are diploid. That means that they contain two sets of chromosomes. So, as humans, we have two different kinds of chromosomes. So, as a diploid organism, we have a total of 46 chromosomes, basically two pairs of 23. So all our somatic cells are going to have that. Germ stem cells are somatic cells, and germ stem cells are going to provide germ cells, which are going to be the sperm and the egg. In order to generate cells that are now only has half the amount of DNA, basically they're haploid, so they only have one complete set of chromosomes. And that process in the germ stem cells will happen through meiosis. So in this image, what we have are the somatic cells and the germline cells, which are the germ stem cells that are going to be generating the haploid egg and haploid sperms. So on the right, you can see a pair of chromosomes from the diploid cells, and you can see that now the haploid egg and the haploid sperm only have one set of those chromosomes. Eventually, a haploid egg and a haploid sperm will fertilize and generate a diploid zygote. The diploid zygote will have one pair of chromosomes from the mother and one pair of chromosomes from the father, making it now again diploid. Through mitosis, you're going to have now the division of all these diploid cells to generate more somatic cells, maintaining a diploid number of chromosomes in each cell, making that organism diploid. Eventually, during development, you will have again the generation of more germline cells, which will generate germ stem cells, and they later will be able to generate more gamete once the organism reaches sexual maturity. So let's take a look at how the cell is undergoing the special type of mitosis to produce germ cells. And what I have in here in this image is a comparison of the meiosis and mitotic events. So you're going to see how they're similar and how they're different. So the first thing that is going to happen in both meiosis and mitosis is going to be the duplication of DNA. So what you can appreciate here in the left during mitosis, you have a red parental homolog and a black maternal homolog. The cell it's going to go through a similar stage of S phase, and both the parental and maternal homologs will be duplicated, creating a mitotic chromosome that has a pair of sister chromatids attached to another through the centromere. Now, these chromosomes are going to pair by type. And the cell is going to have an initial set of anaphase, anaphase 1, which is going to separate these sister chromatids away from one another. Eventually, after the first cell division, you have a cell that contains a pair of sister chromatids. Later, that cell is going to undergo a second set of cell division in which the sister chromatids are going to be separated from one another. At the end, each independent gamete is going to contain only one chromosome, making them haploid. One event that happens when the homologous chromosomes pair, it's that they're going to have chromosome crossover. They're going to recombine, and that is going to make them more rich in genetic diversity. And I'm going to show you that image in this process. Similarly, on the right-hand side, when you now look at mitosis, mitosis is going to go through very similar steps. You're going to have the duplication of DNA. But here, instead of aligning the chromosomes in pairs, they're going to line up independently in this mitotic spindle. A mitotic plate, excuse me. So they're going to now separate independently. So when they have only one cycle of anaphase, eventually each cell will get the same amount of DNA, allowing those cells to remain diploid. So let's take a look again, comparing the ploity, or the number of chromosomes in each cell. During mitosis, a cell that has 2n, meaning that it's diploid, is going to undergo chromosome duplication, so the DNA will be duplicated, so you're going to have 4n amount of DNA, and during cell division, each daughter cell will receive half that DNA, and therefore now you'll end up having 2 diploid daughter cells. In meiosis, you're going to have a similar duplication of the germline cell, generating a 4n intermediate. You're going to have an initial cell division that is going to generate 2 diploid cells, and then you're going to have a second round of cell division that is going to eventually generate 4 haploid cells. It's interesting to know that during spermatogenesis, you will generate 4 different sperms from one single sperm germ cell. But for oogenesis, you generate only 1 cell and 3 polar bodies, which are not cells. That's a genetic material that will be discarded. So only 1 cell will inherit all the cytoplasmic material in the generation of the egg. So now here is a comparison of the way that the chromosomes assemble in meiosis and mitosis. So on A, what you have is the alignment along the metaphase plate of the parental and maternal chromosomes. So in blue, we have the parental chromosomes, P1 and P2, and on red and pink, we have the maternal chromosomes, N1 and M2. So what you can appreciate, they align perfectly in the middle in metaphase. However, during the first division in meiosis, the duplicated homologous chromosomes pair and then they align. So the maternal 1 and parental 1 chromosome pair together, and the paternal 2 and the maternal 2 chromosome pair together. And therefore, when anaphase happens during mitosis, each daughter cell will inherit a sister chromatid from each of the paternal and maternal pairs. But during meiosis 1, the cells that are going to inherit DNA will inherit only one of the homologous pairs, either one maternal chromosome in the left or one paternal chromosome in the right. And that is going to be randomly generated. Now, let's take a look at what happens, as I mentioned to you, when the chromosomes align together during meiosis. And what we're going to see is a physical exchange of genetic information through a process called crossover. So what we have over here is that the duplicated sister chromatids are going to be next to one another, and you can create a crossover event called a chiasma. Therefore, through homologous recombination, like shown in the chromosome TEM in the middle, you can have multiple places where the chiasma could be happening. In this one image is happening in 3 different locations. So what you can appreciate from this is that information from one chromosome arm is now being shared to the complementary but opposite chromosome arm. So if you follow, for example, the light blue chromosome here in the upper left, so this one that I am marking here with an arrow, you're now going to have a light blue region that's going to be followed by a light pink region that continues going down, and now you're going to have another chiasmata where it's going to now exchange information with the dark blue region. So that chromosome has now increased the variability of the genetic material by having crossover events. On the other hand, the one on the right, it's going to start with a pink region, followed by the light blue region, and that's going to then be having a second chiasmata event where you're going to now get complete information from the red sister chromatid. So the replicated homologous chromosomes, eventually they will be separated. So during metaphase 1 and anaphase 1. So what we have here in this image, it's the pair chromosomes that are having the homologous recombination, so you can appreciate the 2 different chiasmata that are being generated. And again, cohesin molecules are holding the sister chromatids together, and the chiasmata, it's holding the homologous pair together. Eventually, when the kinetochore microtubules begin to pull the chromosomes apart, the chiasmata are going to be elucidated and resolved, and as you can see, notice the color in the arms of each of the chromosomes during anaphase 1. So one of them is going to have the tips of them being blue, and the other one is going to have chromosome exchange that are going to make it red. So you have increased the variability in the genetic information in that chromosome. During the second stage in meiosis, what you're going to now have is a separation of the sister chromatids in the same way that you had it during mitosis. So each kinetochore is going to be binding to kinetochore macrotubules. During the centromere, they're going to be pulled away, and now each of the daughter cells will receive only one of the chromosomes during meiosis 2. So the chromatids are not identical at the end, and therefore the daughter cells are not genetically identical to the germ stem cells that generated them in the beginning. So this concludes this lecture. So I will see you tomorrow, Monday, for class, and we will continue looking at this material for the final. Thank you. Bye-bye.